Hi, it's Miss Vital. This podcast is on the senses and is meant for my AP Biology class. Sensory receptors in your body detect stimuli. That triggers action potentials that go to the central nervous system. The ability to distinguish the type of stimuli, like the smell or sight of a fish, depends on the part of the brain that receives the action potential. Sensory organs like eyes and taste buds contain sensory receptors. Those are specialized cells or neurons that detect stimuli. The receptor cell converts the stimulus to an electrical signal. This change is called sensory transduction. It produces a charge in the membrane potential of the receptor cell. There are 5 general categories of sensory receptors. The first is pain receptors. These are probably found in all animals. They are located in all parts of the human body except the brain. Local regulators affect nearby target cells, increase the pain by sensitizing pain receptors. Aspirin and ibuprofen reduce pain by inhibiting prostaglandins. Thermoreceptors detect heat or cold. Some are deep in the body and monitor the internal blood temperature. The hypothalamus contains your body's thermostat. Mechanoreceptors are stimulated by mechanical energy, things like touch, pressure, motion, or sound. These forces bend or stretch receptor cell membranes, making it more permeable to sodium and potassium. Chemoreceptors are found in your nose and your taste buds. They respond to chemicals in the environment. The fifth is electromagnetic detectors. They detect the energy of various wavelengths, like electricity, magnetism, and light. Fish detect obstacles and prey using the receptors found in their lateral line. Some animals use the Earth's magnetic field to migrate. It is thought that magnetite mineral contains iron in their bodies and is affected by the magnetic fields of the Earth. Photoreceptors in your eyes detect light. There are different types of eyes in nature. Most invertebrates have some type of light detecting organ. The eye cup is the simplest eye. This is an example in a planarian. It detects light and direction but doesn't produce an image. There are 2 types of image forming eyes in invertebrates. Compound eyes are found in insects and crustaceans. They are made of many little light detecting units called Amiditida, and each one has a light focusing lens and photoreceptor cells. It forms a mosaic image, and it's excellent in helping these animals to detect motion. The single lens eye is found in squid, mollusks, jellyfish, marine worms. It acts like a camera, and the iris changes diameter like a shutter. Vertebrates have the single lens eye, therefore we do. The sclera is the outer surface. It is a tough whitish layer of connective tissue. At the front of the eye, the sclera becomes transparent and forms the cornea, which lets light in and helps to focus. The choroid is the pigmented layer. The anterior choroid forms the iris, which gives eye color. It changes size and regulates the size of the pupil. Opening the pupil lets light inside the eye. After going through the pupil, the light passes through the lens. The lens is like a disc, and it's held in place by ligaments. It focuses images on the retina, which is a layer just inside the choroid. It contains photoreceptors. They work because light energy produces an action potential in sensory neurons in the optic nerve, and that is sent to the visual centers of the brain. The fovea is the retina's center of focus. It is highly concentrated with photoreceptors. There are 2 chambers that make up the bulk of the eye. The vitreous humerus in the front of the eye is a large chamber behind the lens, and it's filled with a jelly-like substance. The aqueous humor is a small chamber in the front of the lens. It contains a thicker substance, which is secreted by the ciliary body. That substance supplies nutrients and oxygens to the lens, the iris, and the cornea. The aqueous humor maintains the shape of the eyeball. If ducts of the ciliary body become blocked, fluid builds up, and that is a condition called glaucoma. The conjunctiva is a thin mucous membrane, and it keeps the outside of the eye moist. It lines the eyelid. Tear glands are above the eye and they secrete a salt solution. Blinking spreads it over the eye. It drains into the nasal cavity. And humans are the only animals that cry for emotional reasons. This may have an evolutionary basis in stress reduction. In order to focus, the lens bends the light. In some animals, it moves back and forth, and in mammals, the lens changes shape. Ciliary muscles cause the lens to change its shape. They contract to focus on nearby objects. The lens then becomes thicker and rounder. This is called accommodation. They relax to focus on far objects, and that causes the lens to flatten. The human retina has 125 million rod cells and 6 million cone cells. Cones are stimulated by bright light. They distinguish color. Rods are very sensitive to light. They see dim light, only in shades of gray, however. Rods are used for night vision. An animal that can see well at night has more rods. Eye tests are for visual acuity. An eye chart is used. You stand 20 feet away from the chart and read the lines of letters. If you can read them, you have 20/20 vision. 20/10 is even better. That means you can read letters at 10 feet. At 20 feet. A person with 20/20 can only read at 10 feet. Nearsightedness is also called myopia. The person can't focus on distant objects. The eyeball is longer than normal. The lens can't flatten enough, so glasses are thinner in the middle than the outer edges to adjust this. Farsightedness is also called hyperopia. This is when you can't focus on near objects. The eyeball is shorter. Glasses are thicker in the middle. Presbyopia is a type of farsightedness that develops in older age. Astigmatism is a misshapen lens or cornea. It causes blurred vision. The eye is asymmetrical. Glasses are asymmetrical in a way that compensates for the asymmetry of the eye. The human ear is for hearing and maintaining balance. There are 3 regions. The first is the outer ear. It is made of the pinna, which is the outer structure, and the auditory canal. The pinna and the auditory canal collect sounds and channel them to the eardrum. The eardrum is also called the tympanic membrane. It is a sheet of tissue that separates the outer ear from the middle ear. When sound waves strike it, it vibrates and passes vibrations to 3 small bones in the middle ear. The middle ear has those 3 small bones, the hammer, the anvil, and the stirrup, or they're also called the malleus, the incus, and the stapes. The stirrup or the stapes is connected to the oval window. The oval window is a membrane covering a hole in the skull bone. It passes vibrations to the inner ear. The middle ear also opens into the eustachian tube. That is connected to the pharynx or the throat, and it equalizes pressure between the middle ear and the atmosphere. The inner ear is fluid-filled channels in the bones of the skull. Sound vibrations or movement of the head set that fluid in motion. The cochlea is a long coiled tube with 3 fluid-filled canals. The middle canal contains the organ of Corti. That is a hearing organ. It is an array of hair cells embedded in a basilar membrane. These hair cells are the sensory receptors of the ear. The tectorial membrane of the middle canal touches the hair cells. Sensory neurons synapse with the base of the hair cell and carry action potentials via the auditory nerve to the brain. Fluid-filled structures lie next to the cochlea. There are 3 semicircular canals and two chambers, the utricle and the saccule. The three semicircular canals detect changes in rotation and angular movements of the head. Hair cells at the base of each project into jelly-like mass called a cupula. It moves more slowly than your head, pressing on and bending the hairs, sending action potentials to the brain. Hairs in the utricle and saccule detect the position of the head with respect to gravity. These hairs come in contact with gel that has calcium carbonate particles called otoliths. This heavy material presses on the hairs when the head moves, bending them, sending action potentials to the brain. Motion sickness results from the brain receiving signals from equilibrium receptors in the inner ear that conflict with visual signals from the eyes. When we look at taste and odor, they both involve chemoreceptors. Chemoreceptors are found in the taste buds, which detect molecules in solution, and chemoreceptors in the nose detect airborne molecules. Taste buds are an organization of taste receptors on the tongue. There are sweet, sour, salty, bitter, and umami. Umami is a fairly new taste bud, and it's Japanese for delicious. It detects glutamate, like in MSG, monosodium glutamate, also found in amino acids and meats. Smell is accomplished by olfactory receptors. These are sensory neurons that line the upper portion of the nasal cavity. They send impulses directly to the olfactory bulb of the brain. Cilia extend from the tips of these chemoreceptors into the mucus. Odorous substances bind to receptor proteins on the cilia, which generate action potentials to the brain. Taste buds have more sensitivity in children and teens and diminish with age. This is why we are more picky when we are younger.